All right, the next amino acid on the list is threonine. Threonine is hydroxylic. It has a hydroxyl group. The other amino acid that has the same group is serine. Tyrosine has a hydroxyl, but it's listed as aromatic, so it's a slightly different molecule than serine and threonine. I'll just draw it here. Alpha carbon, carboxyl group on the right. Amino group on the left. And the side chain for threonine is very similar to serine with the hydroxyl group, but now we have the methyl group added to it. And I've previously searched for L-threonine, and we can see it on the right here. N terminus, C terminus, side chain. The side chain here has a single bond, so it is flexible. It can rotate about this axis here. It will kind of reach a stable configuration, so you'll kind of have the methyl group and the hydroxyl group in the opposite direction as the amino acid backbone. But these can be rotated, so you can have the methyl group on this side and the hydroxyl group on this side. So there's kind of two different stable configurations for threonine. So here's threonine in the image here. I was calling this a table before. I suppose it's kind of a table. Threonine, polar uncharged, and like I said, it's very similar to serine. Asparagine and glutamine we'll talk about later, but those are very similar to each other, but very different from serine and threonine. Okay, threonine, three-letter symbol THR, and the one-letter symbol is T. Very intuitive. It has 4 codons. It is essential, so it needs to be included in your diet because you cannot generate it from anything else. It was named after threonic acid, which has a very similar structure. It was the last of the 20 amino acids to be discovered, and it was discovered in 1936. I'm not sure if I mentioned this, but it is a polar molecule. The hydroxyl group will be polarized, giving it hydrophilic characteristics. And we looked at this page when we talked about serine, but this is the serine threonine kinase family. It's a family of class of enzymes in the family of transferases. So it's going to so the kinase doesn't necessarily have a threonine on it, but it's transferring a phosphate group to threonine. Since I drew that when I talked about serine, I'll go ahead and just draw it now. So you'll have a phosphate, this is probably ATP or something. It might not be just a floating around free phosphate. something like this. And through a dehydration reaction, this oxygen here will get removed and be a water molecule. And this, one of these oxygens, I'm not sure if it really matters which one, will form a bond, a covalent bond to the side chain. So threonine is important to have on a protein if an enzyme is going to phosphorylate that protein. I was hoping that there was a picture of threonine with the phosphate attached to it, but there's just a picture of serine in that configuration. So the last thing I want to talk about is just catabolism. With threonine, it's essential, so you can't make it, but you can break it down into to form other molecules. So in this chart here, it's listing a variety of amino acids. They can be glucogenic or ketogenic, or both of them, both glucogenic and ketogenic. So threonine is listed here in a variety of places as being both glucogenic and ketogenic. So it can get broken down and eventually form glucose, or broken down and eventually form something other than glucose, but specifically some kind of ketone. And this is the citric acid cycle here in this image. So you can see threonine can get broken down and form various molecules that are in the cycle.